Hello, everyone. I'm Kevin Davis, publisher of CNN, and welcome to the Precision Medicine virtual event hosted by LabRoots. Our next speaker is Dr. Musa Mhlanga from South Africa, and after a few housekeeping notes, I will introduce Musa. We're very excited to have him presenting. We are pre-recording this particular web presentation for scheduling reasons, so there won't be a live Q&A, but I will come back at the end of Musa's presentation to ask a few quick questions. The Precision Medicine virtual event is hosted by LabRoots, the leading scientific social networking website and provider of virtual events and webinars. CNN is proud and pleased to be the official media partner, and we've also played a role in crafting the program for the entire day today. A few important announcements before we get started. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems during this webcast, you can click the support button, which is on the top right of your presentation window, or submit your problem through the green Q&A box, and somebody will try to assist you as soon as we can. This is an educational webinar, and we're offering free continuing education credits. After the webinar is over, we invite you to click on the CE button located in the bottom left-hand corner of the web page and follow the process for obtaining your credits. And so to introduce our next speaker, Dr. Musa Mhlanga. He is a research professor in the Division of Chemical Systems and Synthetic Biology at the University of Cape Town in the Faculty of Health Sciences in South Africa. He's an American-born cell biologist. He holds a PhD in cell biology and molecular genetics. He trained at the University School of Medicine and also Rockefeller University in the lab of David Ho. He later moved to the Institute of Pasteur in Paris, working in the laboratory of nuclear cell biology on issues such as RNA transport and the tracking of RNA in living cells. Musa moved his lab to South Africa in 2008. Where he joined the Council of Scientific and Industrial Research as research leader of synthetic biology. He heads the laboratory for gene expression and biophysics and holds a joint appointment at the Institute of Molecular Medicine in Lisbon in Portugal. His lab works on gene regulation, host pathogen interactions, single molecule imaging of gene expression, and the development of high-throughput biology techniques for screening in both basic and clinical biology. And the title of his talk today is Bringing Precision Medicine in Oncology to South Africa. And so it's a great pleasure to introduce and welcome Musa Mhlanga. Musa Mhlanga.
Thank you for that really kind introduction, Kevin. And it's a pleasure to participate in this international webinar on precision medicine. We are just beginning our precision medicine efforts here in South Africa. And so this talk is really giving you an overview of the state of oncology in Africa and in South Africa, and how some of the approaches that we would like to bring to the oncology clinical space really try to leapfrog to the state of the art that exists in other parts of the world today. So this initiative, to give a bit of background, is called the Biomedical Translational Research Initiative, and it's a partnership between the Council for Scientific and Industrial Research of South Africa, as well as the University of Cape Town Faculty of Health Sciences or School of Medicine. Now, a few facts about cancer worldwide, which I'm sure don't need to be really over-highlighted to this audience, but it's really among the leading causes of morbidity and mortality worldwide, with over 14 million new cases every year, and at least 8.2 million cancer-related deaths. Nixon announced the war on cancer back in 1972, and at least by these numbers, we're not winning that war. The number of new cases is expected to rise by about 70% over the next 2 decades, and that's really linked to everything from a greater number of people living longer in the world to changes in lifestyle as developing nations become richer and people have much more luxurious lifestyles. The 5 most common sites of cancer diagnosed in 2012 were lung, prostate, colorectal, stomach, and liver cancer in men. And in women, they shared a few of those, so colorectal and lung were shared. However, cervical and breast cancer are really the major causes of cancer in women. 1/3 of all cancer deaths are really a result of behavioral and dietary risk. So this is why we see an expansion of over 70% of cancer cases, and really a lot of those coming from emerging economies where behavioral and dietary risk will rise over this time. These include things like a very high body mass index, so BMI, which is also linked to obesity, will rise, a decline in fruit and vegetable intake, as well as in physical activity as people become more prosperous. And of course, there is this a strong effort around the globe to curtail tobacco usage, but these things still contribute quite heavily to cancers that we see. Interestingly, and something quite germane to the South African landscape, is that about 20% of cancers are caused by viral infections. And this really is quite prevalent in low and middle income countries. South Africa is considered a middle income country. And we have more than 60% of the world's new cases of cancer occurring in Africa, Asia, and Central and South America. So 70% of the burden of cancer going forward is going to come from these regions of the world. And most of the research efforts are really focused outside these regions. So they're really focused in North America and Europe predominantly. One reason why viral infections are a very important consideration is because South Africa has the highest burden of HIV in the world, with over 5 million people who are HIV positive. One of the consequences of that is that South Africa also has a very high number of HIV-related lymphomas, more so than any other country. So this combination of viral infection and cancer is particularly relevant in South Africa. So specific to South Africa, the curation of epidemiological data on cancer in South Africa so far has been quite poor. And that is also for historical reasons, but also because a major focus of the Department of Health in South Africa has been on the 2 predominant epidemics, which has been HIV and TB, with over 5 million people HIV positive in South Africa, and 70% of those people testing positive also for tuberculosis. So 70% of that 5 million are co-infected. A huge focus has been on those individuals. So the health system has really been heavily burdened by this infectious diseases that have really made it difficult to mount an effective response to cancer. However, it's pretty clear that a lot of people in South Africa are succumbing to cancer, and the number of cancer deaths is grossly underreported, as is the number of new cases per year. The majority of individuals in South Africa and in Africa have genetic backgrounds or possess genetic variants of known and unknown significance that have not been previously associated with cancers. So this also makes it very difficult to be able to, for example, do effective BRCA1 testing in women, since the variants that are identified in individuals, in the majority of individuals in South Africa, are really of variants that haven't been seen in say, North America or Europe in genes like BRCA1 and 2. one of the most striking things that happens because of this background and sort of the lack of epidemiological data on cancer in South Africa is that we have very highly divergent outcomes for cancer therapy. And that's broadly speaking in Africa and specific to South Africa as well. And those divergent outcomes will be reflected in the next couple of slides that I'm going to show you. So here we see the geographic distribution of non-communicable diseases, amongst which cancer is one. And it's quite striking that, you know, the death rate from non-communicable diseases in Europe is by far the lowest per 100,000 individuals. And you see in Africa and parts of Asia is highly elevated. And this is from data from the WHO, and sort of underlines the fact that the non-communicable disease burden is really the emerging health scare for emerging economies around the world. If we were to take the cancer portion of those non-communicable diseases, what becomes evident is that the death rates that are for each individual per standardized individual is age standardized death rate are also quite elevated in those three continents. Again, Asia, Africa, and South America. And for breast cancer specifically, what we find is that the incidence of breast cancer seems to be much higher in the developed nations, principally because their mechanisms of collecting epidemiological data are far more advanced. More recent data has shown that the actual incidence in women of breast cancer is quite similar, for example, between South Africa and the United States. So around 10% of women will get breast cancer in the United States, and a similar percentage will become positive for breast cancer during their lifetime in South Africa. So those statistics show that the general incidence of breast cancer in these two populations is similar. However, it seems that the genetic variants that we know are associated with cancer between these two countries on different continents are quite divergent. So the ability to test prophylactically for BRCA1 and 2 variants that are driving cancers in Africa is something that is really, really underdeveloped. So when we look at now we're looking at individual statistics for different cancers that occur in Africa. Again, these statistics are quite out of date, and it shows you, 7-year-old statistics show you the paucity of data that's collected on cancer in Africa. You can see here between males and females that breast cancer for females and cervical cancer are very high, and prostate and liver cancer figure very, very high in males in Africa. And sort of these are the primary reasons why we have cancer in Africa. In South Africa, skin cancer turns out to be one of the highest rates of skin cancer in the world, and that's just behind Australia, which has the #1 incidence of skin cancer. Now, there could be several reasons for that, including the fact that South Africa has one of the highest levels of solar exposure in the world. So the highest is in Chile, and the second highest is in South Africa. If we were going to look at mortality rates, this is where the story of cancer in Africa becomes decidedly more sad. If you look to the left of this graph, you see in North America, the blue bar represents the incidence of cancer, and the red bars, for example, all the way on the extreme left for North America, represents the mortality rate. So if you are a woman in North America, the incidence is 10%, or 90 individuals per 100,000, and the mortality is around 15 to 10%. Now, if you look at one of those asterisks, and say if you look at the second asterisk, which represents Southern Africa, so Southern Africa includes South Africa, Botswana, Mozambique, Namibia, Zimbabwe, Swaziland, and Lesotho, so the extreme southern tip of Africa, there is a complete inversion of those statistics. So you have, first of all, a very underreported incidence, as we've mentioned before, but the mortality rate is extremely high. And so you're looking at perhaps over half of women who have breast cancer in this part of the world will die. And those numbers are really shocking, and this is why the South African government has really tried to catalyze an investment in oncology and within precision medicine to be able to reverse these sorts of trends. So what is precision medicine in oncology? And really what it is it aims to identify the genomic and epigenetic changes and patterns in individuals that may influence therapeutic outcomes. And this type of research can really broaden the number of treatment options for patients whose cancer is not responding to standard therapy. And one of the real important technologies that's been able to bring about precision medicine is genomic information, and that has shaped the development and use of some of the newest cancer treatments, but it's also enabled the stratification of cancer patients into appropriate drugs that could be used to precisely treat specific variants that are associated with specific tumors. And we'll get into that a little more deeply in this slide, where we see examples of precision therapy arising some time ago, about 10, 12 years ago with Gleevec. And Gleevec is a cancer therapy that was specifically designed to inhibit an altered enzyme that is related to chronic myelogenous leukemia, or CML. And basically the testing of this translocation that's associated with CML, the fusion of two genes by a genetic translocation, would be a prerequisite for the prescription of Gleevec. And Gleevec was extremely effective in inhibiting this altered enzyme produced by the fusion of these two genes. And this has resulted in a marked reduction of deaths from CML. Another precision medicine drug, and this is again a sort of a first generation precision medicine drugs, is Herceptin. And Herceptin is a drug that is only prescribed after specific types of diagnosis. And in this case, these diagnoses are related to women whose breast cancer tumors are HER2 positive on their surface. And again, this has resulted in very positive outcomes for women who test positive with a diagnostic for HER2 receptors, and led to a marked reduction in mortality of breast cancer, as we showed a couple of slides before. So lung cancer patient tumors can also be treated with precision medicine approaches, and these really center around mutations that exist in the EGF receptor. And those respond positively to two drugs, Iressa and Tarceva, and they specifically target that mutation. And again, genomic information is required to be able to stratify individuals who have lung cancer for their susceptibility to these two drugs. And also, finally, colon cancer sort of falls into this group of first generation precision medicine therapies. And here, mutations in KRAS will get some benefit from the use of these drugs, which are really this Vectibix and Erbitux, which are two drugs that have been used for KRAS mutations. Now, this generation of drugs really points to something quite interesting, which is called kind of development of what is generally thought of as gene-drug association. So we have a gene, and we've identified specific genomic lesions, and these lesions result in the production of aberrant proteins, which can be directly treated with these small molecules in all of these cases, which are able to eliminate the activity of these aberrant genes. There's also a couple of these, like Herceptin, which work by monoclonal antibody, but in general, this generation of drugs, which were associated with specific genetic lesions, are small molecule drugs. The latest precision therapies are more around trying to boost the immune system to respond positively to cancer. And a whole class of these has arisen and has caused a great deal of excitement in the cancer therapy field. And these are termed checkpoint therapies. Currently, only one of these has been FDA approved, and this is the PD-1 or PD-L1 targeting drugs. And this drug is traded under two names, one from Bristol Myers Squibb and the other one from Merck, which is Keytruda and Opdivo. And they essentially use humanized antibodies, which are targeting the PD-1 receptor, which is displayed predominantly on tumor cells. And this receptor is targeted by the monoclonal antibodies, and is then able, by targeting this receptor, is able to mask its presence to the immune system, and that allows the immune system to then respond positively to the tumor. So these drugs really function best, again, when they are accompanied by a companion diagnostic. And actually, this PD-1 antibody therapies were one of the first instances where a drug was also approved with its companion diagnostic that went along with being able to, again, individually target patients for the use of this specific cancer drug. So we essentially now in precision medicine oncology are dealing with the type of genomic information that will really drive research strategies and other strategies that enable us to identify a given set of genomic lesions. And the real power is to be able to identify the markers of the disease, to be able to stratify patients, but not only that, then using, say, immunotherapy to selectively target those markers and use those approaches to become an integral part of precision medicine. And one of the key parts of this is that since we are using genetics to be able to come up with these markers, to be able to target individuals and stratify individuals who have cancer, and use those markers as companion diagnostics to be able to prescribe therapy, then our knowledge of genetic variants becomes very important. And since genetic variance is highly deterministic of our therapeutic approach, the population in which these oncology drugs are developed necessarily plays a critical role in determining the gene-drug associations. Now, that means that in populations like that in sub-Saharan Africa, where there's a high degree of genetic variation and genetic diversity, that becomes important in the development of oncology therapies, and more specifically in precision medicine. Now, since this part of the world has some of the greatest amount of genetic diversity, you would think that it would feature prominently in oncology drug trials to be able to establish the genetic variants and relate that genetic variants to the adequate or correct drug. Unfortunately, that isn't the case, and in fact, the smallest cohort of individuals come from Africa that are used in these oncology therapeutic studies. So there's really a very high paucity of information about genetic variants and its relationship to drug associations. So we know that in looking at genome-wide SNP data from over 100 different populations around the world, and just comparing their genes and their specific geography, we find that there's a very, very strong linkage between the genetic diversity of a specific population and their geography. To put it another way, people who live in the same geography tend to share the same variants. And so since people in, say, in Japan share a lot of the same geography on an island, their genetic variants are going to be highly shared. That means that certain drug regimes and the identification of certain genetic variants that are linked to specific therapeutic outcomes or linked to specific uses of certain drugs is also very likely to be something that has to be developed within the locale of these geographies. Therefore, it means that these sort of gene-drug association studies, which are really the key to precision medicine, will need to be, by default, conducted in these local geographies and take into account the landscape of genetic variation. And this is really what germinates the strong interest in precision medicine as it relates to oncology in South Africa. Because even though specific therapies do exist, the real challenge for using these pre-existing therapies that we know are targeting specific genetic lesions is to understand whether the variants that exist in Southern Africa which are quite different from those that exist in individuals in Europe and North America, those variants in Africa, are they linkable to specific drugs that are currently used outside Africa for this type of oncology? And that's a very important consideration because it's not that the drugs will not work, it's trying to understand the appropriate variants to stratify individuals, to find individuals who would respond positively to those drugs. So the most frequent cancer among women, and this is really breast cancer, and the rate of breast cancer, again, our statistics are not that good in South Africa at the moment, but the incidence is seen to be about 27 in 100,000, with a mortality rate as recently as 2013 of around 3,000 deaths. Mortality from breast cancer accounts for 16% of all cancer deaths in women, and because of all the factors that I mentioned before, women usually present at quite a late stage of the disease. So this breast cancer is something that is one of our pilot cases that we are pursuing in our precision medicine initiative here in South Africa. And one of the reasons is also because there's a very low level of breast cancer knowledge and awareness in general. And since all diseases really have a molecular etiology with significant differences from individual to individual, one of the key parts of just doing basic breast cancer screening, which is very standard in Europe and North America, is screening that's done on genetic variants. And unfortunately, this type of screening in South Africa, even if it's done, there's not much epidemiological awareness of what variants could be linked to BRCA1 that are in the predominantly African population. So, so this confluence of issues really means that there are important challenges that need to be overcome in bringing this sort of precision medicine to oncology in South Africa. And that's really one of the first things we aim to do here. So one of them is to link the African genomic diversity, and that's really provided by different levels of probing. And here we would call these types of probes genomic and epigenetic approaches combined with antibody stratification approaches. And so what we mean specifically there is being able to just use molecular techniques, for example, to determine triple negative status of breast cancer tumors, and to be able to couple that with the ability of doing next generation sequencing of tumors, and in fact even being able to do single cell sequencing of tumors, and then if necessary combining that with epigenetic approaches, so doing ChIP-seq and other types of epigenetic analyses of these tumors. And that would enable us to develop a better understanding of the genetic diversity associated with tumors, and also allow us to develop very targeted therapeutic regimens. Second aim of this stage of the project is really to be able to create something that exists quite uniquely in Europe, which was established by Hans Clevers, which is to create a living biobank. And the concept of a living biobank is a term that was coined by Hans Clevers, and it's the ability to grow tumor organoids, so kind of take a harvested tumor and cultivate that tumor ex vivo into organoids. And by being able to do that, it creates a resource that will be available to scientists, oncologists, and including pharma and biotech companies that can also use that resource to screen for new drugs, for scientists to understand the etiology and mechanisms associated with the tumor, and to be able to interact with oncologists to understand how to develop precision medicine or guided therapeutic approaches to those tumors. So the idea is to use the data generated from this initiative to introduce precision medicine on a much broader scale, and to come up with highly specialized treatments that are adjusted to companion diagnostics. And this is really to reverse this horrific trend where in general in Europe and North America 70% of people will survive cancer, and in Africa 70% of people will unfortunately die of cancer if they get it. So being able to reverse this trend means the ability to rapidly understand and diagnose cancer very early, understand the variants that are linked to the most positive therapeutic outcomes, and those variants that respond most appropriately to precision drugs that can be used, and give patients a very high probability of success. Another initiative we have here is to be able to populate and curate large-scale databases on this sort of information that extend information on South African cancer genomics and therapeutic outcomes. We strongly believe that driving this initiative in South Africa will have major implications for the treatment of cancer in other parts of the world, in other parts of Africa, sorry, and also in other parts of the world. So kind of summarizing our approach and sort of borrowing a little bit from what's being done at the University of Utrecht, where Hans Clevers has established these living biobanks, is really, first of all, being routinely able to take normal and tumor tissue from a patient and take this tissue and be able to sequence it using next generation approaches, and then being able to cultivate the normal and tumor tissues and organoids, and to be able to biobank and store that. The idea, as mentioned earlier, and we're kind of in the pilot phase of this, is to do really routine ex vivo cultivation of these tumor organoids and bank them over the long term. Secondly, we'd like to establish the genomic and epigenetic analyses associated with a subset of these tumors that would inform, of course, research purposes, but also that would enable us in being able to do stratification by immunological phenotyping. So using, for example, antibodies to HER2 receptors and being able to score for triple negative status in breast cancer. And finally, to identify genetic variants of tumors that are linked to specific outcomes. A third reason is we are now establishing a whole drug array, which I will go into a little bit, where we have all the first-line FDA-approved drugs for oncology, and we would like to expose these tumor organoids to these drugs to at least have some sort of information ex vivo of the susceptibility of the tumor to specific drug regimens, to assist oncologists in being able to make informed choices around therapy. So in one part of this, we take the genomic information and subject that to specific pharmacogenomic analyses, and we work with a team of bioinformaticians here who enable us to be able to use existing databases and see whether or not through their pipelines we can infer things, even with variants of unknown significance, whether we can actually elevate those variants to variants that are likely targets or indicators of specific therapeutic options. The second is to really expand tumor cells in vitro culture by organoid approaches, and obviously continuously validate the expansion of those organoids through sequencing methods and maintain those in databases. But in the second part of this, we are really wanting to check drug sensitivity, and something that's not done at all in South Africa. And that allows us to be able to interrogate accessible drugs for their ability to reduce tumor growth and to continuously have that resource available for either new drugs or the use of RNAi or CRISPR interference technologies that are able to target specific genes that could be related to the expansion of those tumors. So on one side, this would help us to advise the oncologists on clinical approaches and clinical treatment. And on the other side, in the cases where we may have much more advanced late-phase cases of cancer, we would use more novel approaches such as array genome-wide screening, or even late-stage phase 3 drugs to be able to understand if there are new and novel therapies out there that could be available to treat these tumors. So overall, this is the workflow that we are currently developing here, and we've been working quite extensively with some leading institutes in cancer research, including the MD Anderson Cancer Research Center in Texas. So another private partnership that we have been working quite a bit with is a partnership that was a small company that came from the CSIR, and that has been able to develop arraying technologies that allow us to rapidly interrogate through RNA interference screening, and also through the use of small molecule treatments or small molecule FDA-approved drugs that will be printed on these sorts of arrays. And that allows us to actually use drug arraying or RNA interference or even CRISPR-I, arrayed CRISPR libraries, to be able to rapidly test for the susceptibility of tumors to specific drugs or genetic interventions. So a lot of these markers of disease that we identify in the first stage of this will be used to develop what we would call knowledge-driven and very marker-specific diagnostics. And we have here at the medical school large proteomics and metabolomics platforms that we are establishing that can confirm the validity of these markers and help to identify them. The therapeutic side is really a secondary side to this initiative, but our idea is really to be able to identify specific substrates and key interacting components, signal transduction pathways, and generate good preclinical validation of these products. We also have just developed a humanized mouse facility here at the university that allows us to do in vivo animal studies using transgenic and humanized mice. And finally, you know, the thing that's really attracted a lot of attention, as I said earlier, is the promise of immunotherapy. And one of the things that we are trying to do here is really to find ways where everybody in the field is trying to do, to be able to stratify patients to groups that would have positive outcomes in to current immunotherapeutic approaches. So, you know, these immunotherapy approaches really have 4 categories, and one of them is adoptive. Some are antibody-based, like the one that I mentioned with PD-1 therapy. Cancer vaccine therapy, like the type espoused by Schreiber, and of course, it's like allergen-specific immunotherapies. All of these are very, very cutting-edge technologies. We have focused a lot on antibody-based immunotherapies in our collaborations, and we're really looking to expand our knowledge outside these to different areas in the immunotherapy space. So just in this last slide, I really want to sort of summarize how we see the process of implementing precision medicine in this space in South Africa, and the reason that we're kind of very, very positive about this is primarily because we want to concentrate the knowledge that we have in this area. We want to be able to get to a state where we have very well molecularly characterized tumors, which is really the state of the art of oncology around the world. We'd like to have very good correlates of intracellular and extracellular targets of a specific tumor or cancer. We'd like to correlate cell surface receptor profile to genomics. So in other words, to be able to know a specific genetic lesion is leading to a change in cell surface receptors. We'd like to know that epigenetic status is leading to a change in, say, metastatic status. We'd like to know that we can validate these markers at the surface. And we'd like to correlate, most importantly, as part of precision medicine, drug sensitivity to the molecular characteristics of the tumor. And we believe that this last one, being able to do this, will really change our awful ratio of 70% mortality in Africa to one that's nearing or approaching what you'd find in Europe and the United States, which is 70% survival from cancer. So with that, I just want to acknowledge my co-principal investigator on this, who is Professor Stefan Barth, who's South African Research Chair in Cancer Biotechnology, who's also here at the University of Cape Town, members of his lab and our lab, and we received large support for this effort from the Medical Research Council of South Africa, the Department of Science and Technology, the Council for Scientific and Industrial Research, and the University of Cape Town Faculty of Medicine. Thanks a lot for your attention. Thanks a lot for your attention.
Musa, thank you so much for that fascinating presentation on bringing precision medicine in oncology to South Africa. I've got a few questions for you, and since I'll take the liberty of asking those, since we don't have the ability for a live Q&A for this particular session. So let's begin by talking about some of the other topics that will be presented here at the inaugural Precision Medicine virtual event. Including a discussion of the Precision Medicine Initiative, and I think also relevant in your presentation, the even more recently announced Cancer Moonshot, as so-called. We have a lot of these big science initiatives, it seems, at the moment, and people are generally excited about them, although there are often concerns raised about the diversion of funds to these big science initiatives that may take away from smaller independent investigators. I wonder, Musa, do you find these, the PMI and the Cancer Moonshots, do these have immediate implications and benefits for the kinds of research and clinical progress you're trying to implement in South Africa? Or is there still a bit of a disconnect, and are there ways that you would like to get more attention from investigators in say, in North America to consider your needs as these programs develop?
Thanks, Kevin. Really interesting question you asked there. I find that the state of cancer therapy and the state of cancer research are really on, I think it's very obvious from some of the slides I showed in this talk, are really on two completely different tracks between Africa and, say, North America and Europe. And so in terms of the Cancer Moonshot, I think we've seen some really exciting developments in cancer research from the laboratory, for example, of James Allison and others that have shown really amazing feats of reversals and remissions that can occur even in late-stage cancers. And the Cancer Moonshot, I think, in sort of, as much as it's a big science project that sucks up a lot of money, I think is going to extend these quite greatly. What I also see is that the National Cancer Institute, which is an institute of the NIH, has taken a very deep interest in cancer in Africa. Most of the focus of the NIH funding to South Africa, the overwhelming focus has been in HIV and TB. The National Cancer Institute recently launched an initiative which our university is hoping to participate in for to kind of elevate the level of cancer knowledge and cancer therapy by developing cancer research centers in Africa. So my answer to your question of attention is that it seems that there is attention focused as well in Africa. Maybe not as much money and maybe not as much visibility, but there definitely is attention that's being focused from the National Cancer Institute. I think also what has happened is that there are a lot of initiatives that are homegrown, like the one that we have been graciously supported by the South African government to sort of kickstart much more interest in this area. But they are encouraging us to work very closely with our colleagues in North America and Europe in basic research and as well as in the pharmaceutical industry. So I think what's emerging here is definitely some deep interest that's coming from North America and Europe. And we may not be at the level of doing TALEN-based CAR therapy, but we certainly are on the radar for other types of immunotherapy like PD-1 therapy.
Thanks, Musa. And I think just time for one final question, perhaps give you a chance to elaborate a little bit on your collaboration with Hans Cleavers, who you mentioned a couple of times. I may have missed it, but what are some of the numbers that you are hoping to aim for in the coming years with regard to developing this South African oncology biobank? And if we could look forward in 3 to 5 years, how do you hope that resource will be assisting in either personalized or precision medicine?
So I'm sorry if I said we are collaborating with Hans Cleavers. We actually are inspired by the fact that they developed this sort of a biobank. We are working with other people at the institute that he heads up. And our real goal is to be able to acquire the same sort of proficiency in developing these living biobanks as they've been able to do at the institute that he heads. I think one of the key things that we'd like to achieve here is the really routine technical proficiency to develop these sorts of living biobanks. So looking out from 5 years from now, as you said, the initiative at Utrecht has been able to, I think at last count, they had over 16 or 17 different organoid lines that they had in their living biobank. And our estimations, as we are not technical experts in developing organoids, would probably be around that same number. I think where we would differ from what's happening in that institute is that our goal is really to be able to do a pilot study that would then be able to show that this was a very good working model that could be rolled out to other parts of South Africa. And so what I would like to see really 5 years from now, less around the living biobanks, because as I mentioned, having a fixed set of them available that's quite representative of the variants, at least in this geography, would be enough. But really what I'd like to see is the process of, first of all, stratification and analysis to be vastly improved. In other words, I'd really like to see that precision medicine throughout South Africa becomes routine in identifying what types of cancer a person has, its genetic etiology, what the best therapeutic options are, and all of this to be done with the latest molecular information and a combination of obviously large databases that we hope we will be able to populate over the next few years about the level of genetic variants that exist for different cancers in South Africa. So the real goal is identification of molecular diversity or genetic diversity and immunostratification for as many people as possible who have cancer in South Africa, for that to be able to inform how therapy is prescribed routinely, and for that to improve outcomes so that we don't have 7 in 10 people succumbing to cancer in South Africa, and to show those trends dropping significantly in 5 years. That would be a positive outcome for this project, for me, and I'm sure for Stefan as well.
Well, Musa, thank you very much. We will leave it there. It's been a fascinating 45 or 50 minutes hearing about advances in precision medicine in South Africa. So that was Musa Mhlanga from the University of Cape Town. I hope you enjoyed that presentation. Continuing education credits are available for those who wish. And this presentation will be archived, like the entire symposium today, for another 6 months. So you'll be able to share links to that presentation and the other 11 or so talks throughout the day with your colleagues and people who are unable to join on the day of the actual broadcast. I think that's all then for me, for Kevin Davis and the Senior Media Group. Our exhibit show floor is open. Please go and support the people who help make this symposium possible, and we hope you will stay tuned for the next presentation here at the inaugural Precision Medicine virtual event. So on behalf of the Senior Media Group and Lab Roots, I'm Kevin Davis. Thanks for watching, and stay tuned for our next presentation at the top of the hour. Bye-bye for now.